everybody. Okay, we will be starting the webinar now. So hello and good morning to everyone. First, I want to sincerely apologize for the postponement of the webinar. And thank you so much for attending. With that being said, welcome to the Open SPR Southeast Asia and Taiwan webinar co-hosted by DKSH and Nicoya Life Sciences. My name is Jim Xu. I'm from DKSH, and I will be co-hosting this webinar for about the next hour. DKSH is a leading scientific instruments and consumables distributor in the Asia Pacific. We're one of the largest providers of biomolecular and biophysical characterization instruments and solutions in the region. And starting this year, we're very happy and proud to represent Nicoya as the exclusive distributor in the countries of Indonesia, Malaysia, Philippines, Singapore, Taiwan, Thailand, and Vietnam. Nicoya Life Sciences is a leading provider of advanced analytical instruments for the biotechnology and pharmaceutical industries. The Canadian company's mission is to improve human life by helping scientists like you succeed. And the topic of today's webinar is going to be surface plasmon resonance analysis of biomolecules with the Open SPR. Okay, so first of all, a little housekeeping. This webinar is being recorded, so you will get the slides and recording via email by the end of this webinar, or a little bit after that. And second, during the course of the webinar, please ask questions in the chat panel to the right, and we will have a Q&A session at the end where we will answer all of your questions then. There is a poll on the right side. There are 3 questions. Please help us answer these questions. They're multiple choice and take you less than 1 minute. And make sure you just scroll through so you can see all 3 questions. And lastly, there will be a post-webinar survey upon checkout. This will help us a lot with improving our webinars in the future, so please help us fill it out. Okay. To introduce our speaker today is going to be Dr. Sharel Figueredo. She is the Director of Field Applications at Nicoya. Dr. Figueredo holds a PhD from UC Irvine and is a seasoned bioanalytical chemist with experiences in protein detection and characterization. She's also an expert in biomarker discovery and quantitation in various different therapeutic areas, and is an expert in bioanalytical tools such as chromatography, capillary electrophoresis, microfluidics, mass spec, immunoassays, and of course, the surface plasmon resonance. And our agenda today is very simple. We will have about an hour-long webinar, starting with this introduction, and then shortly I'll hand it over to Dr. Figueredo, who will take over and give an introduction about Nicoya and the Open SPR, and some selected applications that we feel are hot topics in the Southeast Asia and Taiwan region. We'll also show you an operations video. And after this is done, we'll of course hold the questions and answers session where Dr. Figueredo will be able to answer any of the questions you may have. Okay. So we're very lucky to have Dr. Figueredo here on the call today. I know it is quite late over there in the US now, so thank you very much for agreeing to come join this webinar and speak for us here in Asia. Sharel, can you unmute your mic and say hello?
Hi, can you hear me?
Yes, loud and clear, loud and clear.
Awesome.
Okay, yeah, thank you very much. I will be handing over the control of the slides to you now, so please go ahead.
Okay, I think I have it. Let me see if I can get the pointer. Okay, maybe I cannot see the pointer, but that's fine.
Sharel, how
All right.
about I turn on the pointer for you and hand it back?
That would be nice. Thank you. Ah, I do see it. All right, thank you so much. I can get the point. There we go. So good morning, everyone. I'll be talking to you today about characterization of biomolecules with SPR, and we'll be reviewing applications in immunotherapy and plant-based medicine. As James mentioned, our company, Nicoya Life Sciences, our mission statement is improving human life by helping scientists succeed. A little bit about us. In 2012, our company was founded by Ryan Denome out of the University of Waterloo, where he innovated nanoplasmic biosensors. And we developed our Open SPR instrument and shipped the first Open SPR instrument in 2015. To date, we are over 100 employees. We've installed over 525 instruments worldwide and launched our newest platform, Alto. We are trusted by over 500 researchers worldwide, where they have innovated new and exciting discoveries using our platform. Our customers include academic institutions as well as pharmaceutical companies. Now, surface plasmon resonance provides several advantages. Label-free direct detection of biomolecules in a real-time manner. You get binding affinity and kinetics information such as the rate of association and the rate of dissociation. Now, shown on the left are 4 interactions on this graph, where they have the same binding affinity of 100 nanomolar. However, as you can see, the rate of association and rate of dissociation differ. As you can see, the shape of the curves are different. Now, this information is important as it provides therapeutic performance information in vivo. Now, our nanoplasmonic biosensor consists of golden nanoparticles that are deposited on a glass slide. Light from an LED source creates a sensing field as it passes through these gold particles and collected into an optical detector. We monitor absorbance. Now, this technique is known as localized surface plasmon resonance, as we can monitor and measure binding between a ligand and an analyte very close to the sensor surface. Upon binding, there's a change in molecular mass, which is directly proportional to the absorbance. And so we measure the change in the absorbance resonance peak. Because we use LSPR, we have minimal background interference. As you can see, the optics is very simple yet robust. We have been able to package this into a benchtop instrument that is affordable, easy to use and maintain. Several applications are feasible with SPR. Researchers use it to measure kinetics and affinity. They run competition assays, concentration analysis. They're able to screen a vast library of proteins and antibodies. They can run thermodynamic experiments and even epitope mapping and binning. This can be done with various biomolecular interactions, such as protein-proteins, protein antibodies, antibody-antigen, protein peptide, to name a few. And we are not limited to what you see on the slide. Our Open SPR instrument has been cited in over 100 publications to date in various therapeutic areas such as oncology, immunology, and infectious disease, where researchers are looking at the mechanism of interaction, structure-function relationships, cell signaling, and even discovery of drugs. We'll now review applications in immunotherapy. Now, antibody-based therapeutics have been a central point or central focus in drug discovery and development. Mechanistic studies provide insight into the function of antibody-based therapeutics. Advanced analytical tools have accelerated the characterization of these antibody-based drugs and providing them to be safe and effective. Now, SPR is one such tool that has led to the development of antibody-based drugs and includes strategies such as target identification, ligand modification, cell signaling pathways, and also development of immunomodulatory antibodies such as antibody inhibitors or antibodies that activate new tumor targeting responses. SPR is also being used to screen, as I mentioned, a vast library and vaccine development. So this is some of the work that has been published, and I'll be reviewing these applications. On this slide, we see work done by Gao et al., where they have identified therapeutic antibodies that have a potential against the battle for cancer. They're looking at the ability of tumors that escape the immune attack that severely limits the efficacy of immunotherapy. Now, the mechanism of immune escape that was regulated by GDF15 and inhibits immune cell function is what they were interrogating. So with SPR, they looked at binding kinetics of growth differentiation factor 15, which was bound to a protein called CD44. It's a surface protein on dendritic cells. And this was because this protein was elevated in women with ovarian cancer. So on the left-hand side, you can see the sensogram where GDF15 binds to CD44 with high affinity. And they concluded that this interaction suppressed the inhibitory effect of GDF15, thus facilitating immune escape. On this slide, we're looking at work done by Chen et al., where he's looking at antibody-based strategies to manage sepsis. Now, the protein interaction between high mobility group box 1, that's HMGB1, it mediates sepsis upon binding to tetranectin, inducing immune tolerance and immunosuppression. So they looked at the binding with SPR between the two proteins and whether they immobilized the HMGB1 or the tetranectin. They had similar binding affinities in the nanomolar range, indicating high affinity. They then screened several antibodies, both monoclonal and polyclonal antibodies, against tetranectin and identified monoclonal antibody 8 that bound very tightly with tetranectin. Then in presence of this MAB8, they interacted HMGB1 with tetranectin, looked at evaluated by SPR, and found that this interaction was abolished. This suggested that MAB8 had the ability to disrupt the interaction, and they also tested this in a preclinical setting, and they found that the antibody conferred protection. They concluded that understanding molecular mechanisms was key to developing antibody-based strategies for sepsis. Monoclonal antibodies also are prone to degradation. They're high molecular weight and have high production costs. So this group, Zheng et al., looked at antibody or engineering of antibodies. And so this is an artificial cyclic gamma alpha peptide. And they designed these antibody dimers, M36D, and they showed that it was resistant to enzyme degradation and it inhibited HER2 phosphorylation and tumor growth. So this is a way of developing antibody surrogates for HER2 cancer. And with SPR, they looked at binding between these dimers and HER2, and they showed that M36D could directly interact with HER2 with high affinity and selectivity. They concluded that the strategy of antibody surrogate based on these cyclic gamma alpha peptides could be applied to different receptor target-based diseases. SPR can also be used to conduct various types of screening experiments, so screening from a vast library to identify the most beneficial and efficacious antibody which could be used as a therapeutic. So here we'll see some screening experiments, affinity ranking and epitope binning experiments with SPR. So here we're looking at antibodies for SARS-CoV-2. We have 4 of these antibodies, and with SPR, we've identified and measured the on rate, off rate, the binding affinities, and identified which of these antibodies have the highest binding affinity. We have then affinity ranked them as such, and then carried out epitope binning experiments, which include testing pairs of antibodies against an antigen to identify if they recognize the same epitope or different epitope, and then they are binned based on this interaction. This information, this binning experiments provides information that is important in understanding the mechanism of action or even cell signaling pathway that these antibodies could affect. On this slide, we're looking at work done by Abikaram, where they are looking at PPIs, or protein-protein networks. Now, these protein-protein networks consist of more than 2 proteins. And there's lack of structural information of these protein complexes, and this impedes novel drug discovery. We are looking at the development of antibody inhibitors as therapeutics for a neurosensory disorder called chronic itching. And they're looking at a protein, which is a R7BP protein, which is a G signaling protein. And it binds to a duplex RGS7G beta 5. Now, this protein had a 10-fold lower binding affinity with the duplex RGS7 than for the other duplexes that were tested. The researchers found that by making deletion variants a dominant negative within an N and C terminus, this protein had very high binding affinity. So they then looked at the interaction with SPR between the variant that they engineered and the duplex, and they show with SPR that they have high binding affinities. However, when they test antibody inhibitors, they were able to identify that an R7BP, the protein full length form, was blocked when they identified an antibody inhibitor, suggesting that novel, effective, affordable, and precise anti-itch drugs could be developed with potentially fewer side effects. We can also do direct binding kinetics of antibodies in serum. And here we're looking at SPR binding of the SARS monoclonal antibody, where we have immobilized the spike protein and then titrated the monoclonal antibody. And we're looking at crude samples. So we are comparing the kinetics measurements in buffer, and we find that the data is equivalent between the antibody in buffer and serum. We used a secondary antibody amplification method to improve the limit of detection, and we were able to reach picomolar concentrations. Now, this gives researchers the ability to measure viral antigen and antibody interactions directly in serological samples, and this is key to developing sensitive diagnostic assays. Also, being able to detect the antibody in crude samples, such as plasma or serum, gives physicians or practitioners the ability to look at or evaluate vaccines. They're able to identify which patients may be high risk or may have challenges in their immune response. We'll now go look at some applications in plant-based therapeutics. With the pandemic that engulfed us this past year and a half or so, several researchers pivoted to COVID-19 research and trying to find either cures or drugs that could potentially disrupt the binding of the COVID-19 virus spike protein to the ACE2 receptors on host membranes. Either they repurpose drugs, or in this case, we will look at how plant-based medicines have been instrumental for the treatment of COVID-19. So here we're looking at work done by Gao et al. They used the plant Radix cutellariae, which is a plant used in traditional Chinese medicine, and specifically a molecule known as oroxylene A, a flavonoid found in these roots. They looked at binding between oroxylene A and the ACE2 receptor, and with SPR, they found that this molecule bound strongly or with high micromolar affinity to the ACE2 receptor, potentially blocking the site from viral infection. So the researchers concluded that the investigation of potential antiviral therapeutics could uncover promising candidates to treat COVID-19. Similarly, work done by Hu et al. In researching molecules from this plant, Salvia miltiorrhizia, which is a Chinese sage. They used the molecules salvianolic acid A, B, and C and evaluated binding between these molecules and the spike protein, as well as the ACE receptor. So on the top three figures, you see this is salvianolic acid A, B, and C interacting with the spike protein, and in the bottom figures, we're looking at traces of the molecules as salvianolic acid A, B, and C with the ACE2 receptors. They measured the binding affinities and concluded that salvianolic acid B showed the greatest binding affinity and anti-219 nCoV pseudovirus effect. This provided new insight into the use of traditional Chinese medicine for the treatment and control of COVID-19. Now, all this work has been done on our Open SPR platform. This is the instrument as you see it. It's a benchtop instrument. It's a manual in the sense the researchers are injecting reagents to build the sensor surface and then test binding between a ligand and an analyte. There are two channels, one as a reference and one is the active channel. Now, shown on the right is a scientist lifting out the sensor. You can see the gold nanoparticles in this oval shape deposited on the glass slide. Now, we have various sensor chemistries to help evaluate various biologics or proteins, either using our covalent chemistry, sensor chemistry, or our capture sensor chemistry. We also have plain gold as well as the membrane and lip chip. To allow for automation or high sample size, we've got a autosampler that's fitted with the Open SPR instrument. This is the same two-channel Open SPR instrument. The autosampler can hold 2 96-well plates, and the sample area is cooled to 4 degrees centigrade. Now, the autosampler allows for automated injections from these 96-well plates into the Open SPR and then conducts the SPR analysis in an unattended operation for about 24 hours. So we'll now look at a video as to how the Open SPR works.
So Sharel, I will be giving you control, okay?
Thank you.
To present.
All right. So can you see? Let's go to share. Let's see. Great. Are you able to see my screen?
Not yet.
Oh, no, okay, let me go back and see it.
Yeah, it's starting to share content, so it should appear soon.
Okay.
No worries. Yes, we can see your screen.
Yep, okay. So now you're able to see my video, I'm hoping. Whoops, it didn't open. Hang on, sorry about that. Okay, great. Are you able to see the screen? Open SPR video? All right, thank you. Okay, so we'll now look at the use of the Open SPR. You see how it functions. We have the reagent bottles on the side, and now we are looking at the various compartments within the SPR instrument. Here is your LED source. This is your light source. You then have your flow cell. So this is where from the injection port, reagents are flowing through this flow cell. The sensor chip holder will hold the sensor, and this, when it docks to the flow cell, an SPR experiment can be conducted. Here is your lens. So this is where you have your optics that is going to collect the absorbance that's passed through the sensor chip. We have two spectrophotometers in here, one to collect the trace for the reference channel and one for the active channel. So as you can see here, this is on the side of the instrument. We have 4 reagent bottles. So one usually is the DI water for the rinsing. Then you have choices for your buffers for various parts of the experiment. So these are placed inside the instrument, and now we're ready to set up the sensor. Once you have decided what chemistry you need, let's say you're going to use a carboxyl sensor where you will be utilizing covalent chemistry, you'll then need to start up the instrument, prime it, following the instructions, and then you load the sensor. As you can see, the sensor is really very small. It's just a glass slide. It's rinsed and then placed into the chip holder, which is then loaded inside the Open SPR instrument. And then from then on, the Open SPR software is able to begin the acquisition. We're going to use an immobilization wizard, which is where you are able to choose which sensor chemistry you are going to run. So what's nice here with the ligand wizard, after you've input the various parameters that's required, such as the molecular weight of the ligand and the analyte, the software is able to control the flow rates and the channel to where the reagents need to be added in automatically. Should you not wish to use the ligand wizard, you're able to do this manually as well. Here you're seeing the scientist injecting a cleaning solution, which is loaded it and injected HCl to clean the injection port. And then you're able to see 2 traces, one in red and one in blue. The red is the reference channel, which serves as your blank, and then your active channel, which is where you will immobilize your ligand. And so we are looking at the response. So on the left-hand side of on the y-axis, you can see the response units. We're just looking at signal increase or signal decrease, depending on whether you have association or dissociation occurring. So as the buffer is flowing through, you can see the next reagent that we have loaded is the activation reagent, which is the EDC-NHS, to activate the carboxyl group so that we can immobilize the surface with our ligand. And this is how the protein or the antibody will bind to that sensor. So with the EDC-NHS, you see a rise in signal. And once the EDC NHS has flown off the sensor, then you can see it come off. But you can see a much increased baseline as reagents get added on and built onto the sensor surface. So now the next part is to immobilize our ligand, and this is protein A. The software guides the researcher to add the next reagents. And as you can see, the ligand is immobilized only in channel 2 and not in channel 1. So you can see the reference in red, the baseline is low, whereas you have a nice sharp increase in the active channel, indicating protein A has been immobilized. So using the ligand wizard, the software calculates how much of ligand is necessary and whether you have reached it or not. And once the target has reached, it gives you a little message. However, if the target was not reached, you have the option to inject additional ligand. The next step would be to titrate or add titration points of analyte, which is multiple concentrations. We begin at the lower concentration, and this is occurring in both channel 1 and 2. Any non-specific binding can be observed at this time in the reference channel if there was any. So we will add the names of the analyte that we are adding, and this is going to be IgG. We're beginning at 11 nanomolar concentration. And so you can see you have very specific signal of the antibody that's bound to protein A compared to the reference channel. You also have the ability here to look at the data in either in the corrected view or the raw signal. You're now beginning, as you can see, once in the corrected view, we are only seeing the corrected data. So no, we don't see the baseline. We then have injected glycine HCl, which is our regeneration solution. This is added to remove that concentration of analyte. And then we look to see that the baseline has been brought down and ready for the next analyte concentration, which is 33 nanomolar. That has been injected. So for all these injections, there's a researcher or scientist that's injecting these reagents into that Open SPR port. If you have the Open SPR XT, this can be done in an automated manner. So the flow rate also, as you can see on the top, is about 40 microliter per minute. You have the ability to change the flow rates, either bring it low or high, depending on the data that you see. You have the ability to change the bottle buffer should your sample require a different buffer. And so we're going to collect now a few data points for IgG. As I mentioned, we run them at various concentrations. And we are looking to see a 5% drop approximately. And then once we have collected that data, this is fast forwarded. So you're able to see that we have a series of injections that are done at various concentrations. And then in between these injections, we have a regeneration solution. So this is a multi-cycle experiment. Once this is completed, we will be able to do an analysis in our software called TraceDrawer. So the sensorgram or the file is imported into this software, and you're looking at the corrected data. Actually, all the data points will be here, where we are now going to deselect all the regeneration solutions or the blocking solutions and only look at the 4 analyte concentrations that we had tested. For post-processing, we first want to ensure that we are selecting the right time points, the right concentrations. And so we are adding the values of concentration based on what we had prepared. And that's what's being entered right now. And then if there's a slight change that's needed to adjust the rate of association and dissociation time points, that can be done. We then select the stoichiometry based on the type of interaction, and that's the fit model. And then when you click on fit, the software is able to calculate the kinetics, which is the Ka, Kd, and the binding affinities. Okay. So with this is the overview of the utilization of the Open SPR instrument and the data analysis. So at this point, I will hand over the controls to James.
Thank you very much, Sharel. Okay. So we will answer all the questions in just a quick moment. And just a quick reminder, please do help fill out the poll on the right. Okay, so thank you, Sharel, for that. It was a really clear talk, and I think the video is very helpful as well. I think it shows how easy it is that we can use the Open SPR to do research. And to make a small conclusion, the Open SPR is a great research tool because it does improve the quality of research for those who are maybe not using a label-free tool by improving the sensitivity and specificity of your binding of protein interaction research. It reduces the complexity because it makes the SPR technique available and easy to use. And it's also simple to learn, and you can really see the hands-on data real time as it comes. And also it's accessible and affordable by enabling it on a benchtop instrument with the new sensor chemistry and with the new detection technique so that any operator can use it at their benchtop. And we will be moving on to the questions and answers part of this webinar. Please do feel free to ask questions. And it looks like we already have one question. Sharel, may we check if we can reuse the probes?
Yes, we can use the sensor chips again. So we have regeneration buffers that remove the analyte, as you saw, that then once the, let's say the first experiment was completed, you can next then test a different analyte. So you can do these, you know, as long as the ligand is stable on the sensor chip, it has not degraded. So as long as the ligand is not come off, you can reuse the sensor. Typically, we would say at least 60 to 80 runs you should be able to do. Again, it would depend on the type of ligand or what type of protein that's used as a ligand.
Okay. And another question is, how would nucleic acid samples, so DNA, RNA, work with this, with the Open SPR?
So what we suggest is either usually biotinylating the nucleic acid, whether it's your DNA or RNA or aptamer even, and that can be then used on our streptavidin sensor chips. So we would immobilize it that way, and then you can test the interaction either with an additional RNA or DNA or protein molecule, and that's your analyte.
Okay, thank you. And another question. Hang on.
Yep, I see the question.
Yeah, the question is, how about the analysis of nanoparticles such as exosome? We only can determine the Ka and Kd of the ligand present on nanoparticle. And can we characterize the nanoparticle in terms of size and quantity as well?
So again, it depends on the size of the nanoparticle. We have already worked on nanobodies, for example. It depends on the size of the exosome or the nanoparticle, if the And I'm thinking if it's within the 40 nanometers, because this is the length of our sensing field, then we may be able to do just a yes/no binding. However, we have worked with and been successful with nanobodies. So it would again depend on that size. So it is a possibility. So you just need to have your ligand immobilized, and then your analyte could be your exosome. You would get a yes/no binding.
Okay, and next question is, what are the advantages of the Open SPR as compared to traditional SPR? I think this is the favorite question.
Yes. So, traditional SPR has the sensing field is much higher because it's not gold nanoparticles, but it's a gold film that they use. So that sensing field is longer. What this means is that it most likely when analyte is flowing, you're not measuring the specific binding between the ligand and analyte. You might be also getting bulk effects, which means analyte in the solution that's flowing, not necessarily binding, could also lend to binding. And this could skew your measurement. With SPR, because the length is of the sensing field is much shorter, about 40 nanometers, you're able to get specific binding between the ligand and an analyte. And so you minimize your bulk effect. So this is one big advantage. The other things are the optics, for example, that you need. And then, for example, with traditional SPR, it uses like prisms, lasers, and they are prone to artifacts as well as vibrational changes. So these are some things to keep in mind. The other thing I also know is that it really requires a lot of skilled personnel to open or use the traditional SPR instrument. So unlike the Open SPR, which is very easy to use, any scientist, whether it's a grad student, a postdoc, anybody can use that instrument. You don't need specific skills to use the machine. We would teach our users assay development and optimization, and that would be the chemistry is what would be needed to, we would need to train them on.
And Sharel, just to follow up on that question from myself, in terms of the applications of traditional SPR or compared to the Open SPR, would it be the same set of applications? So for somebody evaluating the Open SPR, would it be correct to say it's the same set of applications and possibilities that you can do with the traditional SPR?
I would say we could do most applications. The one that is challenging would be small molecules, just because that field itself is challenging for whether it's traditional or Open SPR. So keeping that in mind, I would say it would depend on the size, the ratio, and the chemistry, will it work or not? But most applications like proteins, I said protein antibodies, antibody antigen, these and biologics, looking at structure function. So these are very similar to the traditional SPR applications.
Okay, thank you, Sharel. Another question, what is the benefit of having kinetics on top of your KD value?
I had mentioned that in my talk, which is that you get the on rates and the off rates. So the K on, or the rate of association, which is the time or how the therapeutic would bind onto the, let's say, your ligand. So that's the time that's interacting. And the off rate really talks about stability of your therapeutic. So when you're looking at binding, especially you're looking more in therapeutic areas to make medicine. So this is really important, this information, as it gives you insight into how these drugs would behave in vivo. So when it's given to either animals or before it becomes a drug, then you would test it on human beings then. But so this is the information is critical for therapeutic performance information.
Okay, thank you, Sharel. Do we have any more questions? Okay, if there are no more questions, then we will move on from the questions and answers. Okay, so just a little bit at the end. So thank you, Sharel, for answering the questions, and thank you to the people who asked the questions. That was very helpful. And for the very last bit, I want to give a small introduction to DKSH for those who are not familiar with us. So DKSH is a market expansions services company serving the Asia Pacific market. And our strengths are particularly in biophysical and biomolecular characterizations, material characterizations, chemicals, and in the life science instruments business. So as I mentioned earlier, DKSH is the distributor and partner for Nicoya in Southeast Asia and Taiwan, which if you are in this webinar, hopefully you are part of this region. And if you have any further inquiries about the Open SPR, please do get in contact with one of our executives or colleagues shown on this page. So you'll have the corresponding email for the corresponding regions. And we would be happy to work with you and work with Nicoya to help you evaluate whether the Open SPR is right for you. Lastly, I want to give thanks to Dr. Figueredo for your time again today. I really appreciate that you took your time in the evening to promote these applications and immunotherapy and in plant-based drugs to us. And I think it was some great examples, an overview of what can be done and how SPR is useful in some of the local research that we're doing in the region. I also want to give thanks to Irene Chen for coordinating this webinar. Irene's sitting next to me, so thank you very much, Irene. And lastly, thank you to the audience for attending. Again, just to apologize for the rescheduling, so you had to basically come out twice for this webinar in terms of timing, so really appreciate you for coming on board. I hope it's been useful for you, and hopefully you've seen how surface plasmon resonance could be useful, how this technology could fit on your own benchtop and help you accelerate your research and help you in your workflow in whichever molecule or therapeutic area that you are working on. And just to follow up on the housekeeping, we will be sending the recorded webinar to you and the slides over to you a little bit after the end of this webinar, so you can review it. We will be ending the webinar now, so thank you for your time and have a wonderful rest of your day. And thank you, Sharel, so much.
Thank you, James. Thank you, Irene. I really appreciate you hosting Nicoya and inviting me to speak. Thank you so much.
Yeah, it's great to have you. Bye-bye.
Thank you. Bye-bye.